I know a lot of you are interested in ivermectin for cancer treatment, and there is a clinical trial ongoing right now in California, and some of the latest results have come out. They're so fresh that there are still not actually published results. It's a form of a conference abstract, but it's giving us some preliminary early information why this group of clinicians even considered using ivermectin for cancer treatment, and it's a specific type of cancer treatment. So ivermectin is used in combination with another medication, which is a immune checkpoint inhibitor. One of those, there's many, but specifically it's a immune checkpoint inhibitor that it's an antibody that targets a molecule called PD-1. Check out my past videos on this topic, specifically the video where I recently made that discussed the idea that mRNA vaccines can also sensitize cancers to that very specific medication. So, and I explain in detail how that works, so you can check that for the background. But the take-home message here is that these medications, what they do is they prevent interaction between cancer cells and T cells, and that interaction, when that interaction happens, it prevents T cells from being able to kill the cancer cells. And then by introducing an antibody that will target this PD-1 molecule that can be on T cells, it prevents that interactions, and then T cells are free once again to start destroying cancers. So that's the premise behind that. And one of the clinicians involved in this clinical trial, I believe that he was involved or spearheading the research that discovered how ivermectin might play an important role in helping sensitize cancers to that very specific medication. And I'm going to tell you about that first, because that publication, when it came out, made a lot of noise. It was incredible research and new information about ivermectin that opened the eyes. And the cool thing is that the authors of that article mentioned at the very end that looks very promising based on work on mice, and it suggests that ivermectin can make cold tumors hot. Again, check out that video on how mRNA vaccines can make cold tumors hot as well, and therefore sensitize cancers for that medication, to get the background on it. And this should be tested in clinical trials, and voila, these guys, for once, we ended up having a clinical trial, and one of the authors is involved in that clinical trials. I believe that's Dr. Peter Lee. For once, I remember the name because it's a little bit easy to remember, but this guy is a renowned oncologist that, as far as I understand, has been interested in immunology of tumor sites, of what happens to the immune system, which is very complicated information. And little by little, this guy has been discovering what is going on over there, what's happening, and learning about immune checkpoint inhibitors and why they work or not work in the cancer setting. And in the past, I believe he was involved in wanting to discover, hey, is there a way that we can bring additional drugs that could sensitize cancers to immune checkpoint inhibitors? And when they were screening these drugs, ivermectin worked amazingly. And that prompted them to test this theory on animals. That's how it normally progression of research. First, you go drugs on cell lines, then you go to the animals, and then you move on to humans, right? And let me now break the paper down for you. Oh, by the way, my name is Dr. Mikola Rashek of Merigenomics. Let's get started. So, as I mentioned, this paper came out in 2021. I believe it's the same group of authors that discovered how ivermectin might be sensitizing cancer cells to immune cells, specifically to killer T cells. I'm not going to get into the details, but one of the ways that it does that, it causes a type of cell death that is immunogenic, meaning normally when a cell dies, the body just cleans it up and doesn't stimulate the immune system. And there are reasons for that, because obviously we don't want to be doing this. We have many cells die, but you can also have a cell death that can stimulate the immune system's response to it. And when it absorbs the fragments of the dead cell, such as cancer cell, it will be monitoring what it actually is interacting with and start responding to the fragments that are discovered, because they are cancer associated fragments or molecules, right? So there's this immunogenic type of cell death, and it turned out that ivermectin can cause that type of cell death in cancer cells. I will not get into the background right now as to why that happens, how ivermectin actually achieves that. I will perhaps do that in another video, because it's just complicated. But that's one of the ways that it does that, and the outcome of that is twofold. It sensitizes the cancer towards killer T cells, or effector T cells, you can also call it. Those are the type of T cells that are supposed to come in there and start killing, destroying the thing, while at the same time it starts to remove immune cells that would normally be suppressing the immune system. And some of these can include regulatory T cells. Again, check out my past videos where I explain regulatory T cells in a context of when I was explaining how ivermectin works against parasites, and how such fight against parasites might inadvertently or indirectly help fight cancer cells via regulation of these regulatory T cells, because regulatory T cells are the type of immune cells that are supposed to come in at the end of the fight and clean up, make sure that the immune system doesn't continue doing its work, so they are suppressing the immune system. But cancer cells can take advantage of that sometimes, attract these cells to the cancer environment, and use these cells to help them not be recognized by the immune system in order to be killed or attacked. So that's what ivermectin can do. Can stimulate these killer T cells to go in and destroy things, while at the same time it can suppress those regulatory T cells that normally would be helping cancers, but obviously we don't want that. So that's the magic trick. So what did these guys do? They wanted to test this in mice. So when you cause this immunogenic type of cell death, I don't remember exactly what it's called, but this immune stimulating cell death, there's certain molecules that are released. One of them is HMGB1. The name is not that important. It's a molecule that is inside nucleus, and it organizes the DNA to make sure that it's organized properly. But once it's released from the nucleus and it goes outside, it is a marker for the immune system that something is wrong, and the immune system should come in there and check this out. So that's a hallmark of that type of cell death. Another one is ATP is released. So ATP think of like those are energy molecules that we need for all of our cells to do some work. So I think of ATP as miniature batteries. So they are used by our cells to basically perform all the functions. They store energy that can be then used by the cells to do all the work, and this is how we live. And then there's also released a protein called calreticulin, I believe. I hope I'm pronouncing it correctly. But again, that protein is in a specific compartment of the cell, and it's involved in making sure that the cells are folded properly. That one is not released outside the cell. It remains attached to the cell, but it can be outside the cell. And again, it tells the immune system, whoa, something's seriously wrong. come check this out and start destroying these things. So those are the hallmarks. And these guys wanted to test ivermectin in mice. And what they did is the following: they took mice, and they were injecting them with specific breast tumor cell line. Why breast? Is because they were specifically interested in triple negative breast cancers, and the reason why is because that was of interest to this group to eventually treat via ivermectin and those immune checkpoint inhibitors, and this is exactly what they're currently testing in that clinical trial. So they injected these mice with these cancer cell lines, and that results in of course tumors. And what they did is they checked the release of that nuclear protein, and they used a green fluorescent antibody. So that means an antibody recognizes this protein, and wherever it recognizes, it attaches it on the slide, you will see this green fluorescence. But when they added ivermectin along the way, you saw a reduction of this fluorescence. And what that means is that the reason why is because in the process of washing, remember that protein was released outside the cell. In the process of washing, when you bind the antibody, you need to wash some of the antibody away, because otherwise everything would fluoresce, right? So you need to wash the unbound antibody away. When you do that, the protein that are outside the cells are washed away. Why is this? Because when you are preparing cells or tissues for examination, you fix them first. So you add a specific chemical called formaldehyde, and formaldehyde allows to cross-link everything, meaning there is a chemical bond that starts occurring between proteins and DNA, and they all interacting with each other and creates this one giant immobile mesh. It's in a way an ability to freeze everything in space and time. So this is why we do this. So it's that specific way of making sure these proteins are stuck in place, and then it allows you to monitor where they are within the cell. And if the protein was outside the cell, then of course they're not captured by this mesh that you created, and in the process of washing, it will be washed away. And this is exactly what they showed, that when the ivermectin was being used on these mice as well, there was less of that fluorescence. And the reason what that meant is that ivermectin was causing that cell death in those tumors that they were examining here. By the way, in pathology, in hospitals, etc., when you're looking at all these tissues that clinicians are looking, or pathologists are looking at, besides formaldehyde being used in order to fix everything in place, like I just told you, these tissues are also embedded in paraffin, and that's basically a wax. So why wax? Is because once you prep it all in wax on top of that, then you can slice it super thinly and look under microscopes, and this is how pathologists can see, hey, what they are seeing under the microscopes, and recognize abnormal cellular behavior, and recognize this is what's happening, there's cancers going on here, and so on. So that's the, and this is called formaldehyde fixed paraffin embedded process, right? And those are the tissues. Whenever people, and just a side note, whenever people then test these for under microscope for spike protein, and they claim, hey, vaccine spike protein is present, they never actually prove that this is vaccine spike protein. Why is that? Because in order to prove that, you would have to check the amino acid sequence of the spike protein. So you'd actually have to see what is this spike protein made of, because vaccine spike proteins had specific amino acids that are not present in the virus, but that has never done that. And the reason why is because you would have to use complicated chemicals to get rid of that wax and get rid of that formaldehyde in order to free the protein. Then and then you'd have to isolate the spike protein before you can analyze its spike protein sequence. So as far as I know, nobody has ever done that and published yet in order to show that indeed truly vaccine spike proteins have ever been discovered in any of these biopsies that have been discussed in scientific literature. They mention, oh yeah, this is vaccine spike protein, but it's not actually proven ever. Okay, so that's one way to prove it. So that was one thing. So they looked for that nuclear protein, and with ivermectin treatment, that nuclear protein, there was less of it, indicating that it was washed away because it was thrown outside the broken cells. The reason why, because ivermectin caused cancer cell death. Now, another thing they looked at is calreticulin, that special protein that helps proteins fold properly. That's one of its functions. It has other functions as well. And it's funny, there are mountains behind me and they're gone behind clouds. And what happens is that one, remember I told you, it still is retained on the surface of dead cells. And so here, when they were looking for calreticulin with green fluorescent antibody, you see the opposite effect. You see more antibodies attaching to the tumor because more tumor cells died and more calreticulin was exposed, but it was still attached to the cells, so therefore it wasn't washed away. So you see more green fluorescence. And in red fluorescence, they were showing the tumor cells themselves. And one more thing they did is they also checked the infiltration of these tumor cells by the T cells, and they looked at CD4 and CD8 T cells. So the CD8 T cells are the killer T cells, or those effector T cells, another name for it. And clearly you can see that they were looking for them with green and yellow color, and you can clearly see that the tumor sites were being infiltrated as well more when ivermectin was being added to the process, right? So clearly showing that ivermectin can help make cold tumor hot, meaning that there is more immune cells in the environment of the tumor cells. And this is what you absolutely need in order for that immune checkpoint inhibitors to function. Why is this? Because if the tumor is cold, meaning it's not even infiltrated by T cells, then of course the medication that is supposed to prevent the interaction between the two tumor cells and say killer T cells, well, of course it's not going to work because killer T cells are not even there. Hence, it's not going to do anything for you. Therefore, when you bring T cells to the tumor and you add that special antibody that prevents cancer cells from stopping killer T cells from doing their job, this is when it's effective. And this is why they were interested whether this works in animals and why they wanted to prove it. So the next thing one more thing that they did was they used an interesting form of vaccine. I'm going to call it vaccine loosely, because what they did is they took mice and they injected them either with just tumor cells, and they were monitoring growth of tumor, or they injected them with tumor cells that were first washed with ivermectin. And when they did that, they could see that such mice, the cancer was not growing as extensively or rapidly as if ivermectin was never used. So again, another proof that they did. They also showed that one of the ways that this was all achieved is because ivermectin was again showing suppression of those regulatory T cells, or T cells that are normally, when they enter tumor site, they can suppress the immune system. So ivermectin was suppressing them, so that's what it was helping them. And the last thing, they also looked at survival of mice after they had induced cancer growth. Oh, and before I get there, I should mention, of course, they also tested the effect of combination therapy as well. So they looked at the tumor sizes as well when they didn't do anything. Tumor size with just ivermectin, tumor size with the anti-PD-1 antibody, so that immune checkpoint inhibitor, as well as a combination. And in all of these instances, the combination with ivermectin and the immune checkpoint inhibitor, that was the best combination that reduced the tumor sizes in these mice. So clearly showing that what they were hoping for, what they were predicting, is indeed working, which is what, like I mentioned, eventually led to the clinical trial we now have with humans. The last thing is that they also checked whether this helps in terms of metastasis, and they did show that indeed it helped with tumor metastasizing as well. This is really cool what they did there too. They took those tumors as well, they took those breast cancer cells that they were injecting into mice, but they made sure that these cells also had a specific protein that would fluoresce if you expose it to specific type of wavelength of light, and that would literally allow them to monitor where the tumors are growing inside mice by literally looking where they see fluorescence in these mice. And so they were able to show that when the mice were injected with the cell lines, they operated on them, they removed tumor, and this is where the issue is that perhaps tumors can come back, and the tumors were coming back. And after tumors started coming back, they treated them, and they again treated them with only ivermectin or only immune checkpoint inhibitor, so that antibody that targets PD-1, or a combination. And they also did one more combo, which was a triple combo, which was ivermectin, that anti-PD-1 antibody, or the immune checkpoint inhibitor, plus interleukin-2, which is a cytokine that stimulates those killer T cells. But the best one was the combination of ivermectin and the immune checkpoint inhibitor. The interleukin-2 addition didn't do much statistically. It made a little bit better, but not much, so it didn't make much of a difference. So all of this data pointed it out too, that hey, ivermectin could be a fantastic additional drug to sensitize cancers to this specific immune checkpoint inhibitors. Why are we so keen on these immune checkpoint inhibitors? Is because when they work, they can have fantastic results, and they can really save people's lives. When without this medication, it's almost a guaranteed death. If I can, I know it sounds tragic, but that's why we're so keen on this medication. Ironically though, this medication has also can in certain instances lead to development of turbo cancers. So turbo cancers are real. It's this specific medication that causes it in some individuals. It's still being discovered as to why that happens, and it's called something else in medicine. It's called hyperprogressive disease, but it is a real thing. And check out my videos on turbo cancers if you want to understand as to why people are suspect that mRNA vaccines might be linked to turbo cancers. And it has to do with the fact that that medication, that antibody, is an IgG4 antibody, and that in itself, under certain circumstances in some individuals, is a contributing factor towards causing this turbo cancer or hyperprogressive disease. So that's a trade-off. But overall, because of the benefit in the vast majority of people that are being treated with this medication is so good, overall, we are still sticking with using this medication, even though there's a small risk in some individuals that what it will do instead is cause cancer that will now be developing so rapidly that it ends up killing those people. And before this medication, this antibody against PD-1 molecules started to be using as a medication, there was never description of turbo cancer. So this is something still fairly new on the medical scene, but it does precede the pandemic. And I explain all of that in my videos dedicated to this topic. All right, this is everything I'm going to tell you today about ivermectin and cancer in this series dedicated to this topic. Before I go, please like, please share, please subscribe if you haven't already, and also please check out my Patreon account for a couple of reasons. Number one, if you're a Patreon member, then I'm working on a project right now where I want to create a guide for what it means to live a healthy living lifestyle. So, and I want a contribution from people. I have my own rules, and I follow these rules religiously, but it doesn't mean they are the best or the smartest. And I want to know what you guys think as well. And I want my patrons, all patrons, whether you're supporting or free patron, will be voting on what that guide should be made of in terms of most important to least important, right? So that's number one. And number two, if you are supporting patron, you also get to vote and determining as to what videos make it to eventually to YouTube, because not all my content migrates from Patreon to YouTube. All right, I'll see you guys next time. Bye, everyone.